slides. I'll start with the slides. Just a moment. So can you see the screen? Yeah, fine. So CRISPR is actually prokaryotic memory of invaders. CRISPR is actually an immunity of bacteria, basically. And it works as a primitive immune system of bacteria. And in nature, it works in this way. But overall, as it was discovered, it has been employed as a genetic engineering tool. So I'll tell you in this talk how all of this happened. So here, what you can see in the picture, just a moment, you see pages invading bacteria. This is the membrane of bacteria, and there are pages on the surface of bacteria invading DNA into bacteria. And bacteria has to fight against phages, and basically it's like an evolutionary arms race between bacteria and phages. And therefore, bacteria developed many different systems to fight against phage infection. One of those systems is CRISPR. And here is actually my introduction slide, just a little bit for you about Lithuania. And this is the flag. This is a picture of the university. This is the life science center we are working in. And this is our group. It's actually quite a big group, 30 people in total. And the head of the department is Professor Virginija Šikšnys. And this is, in the circle is me. And CRISPR. Some slides are actually quite complicated, especially if you are from humanities. But I'll try to be slow. CRISPR actually is an acronym which stands for Clustered Regularly Interspaced Short Palindromic Repeats. What are these repeats? These repeats lie in bacterial DNA, and it's repetitive nucleic acid sequences. And these repeats are interspaced with spacers. And these spacers came from invading nucleic acids from phages. And this is the step which is called spacer acquisition or adaptation. Actually, this CRISPR region sometimes is called like barcode, because in this barcode, you write, bacteria writes down and can memorize invading phage. And by inserting a new spacer, it knows that this phage already invaded bacteria. And after the adaptation stage, comes the stage of gene expression and RNA biogenesis. Because CRISPR is actually RNA-guided machines, RNA-guided proteins. So CRISPR proteins need not only Cas genes, which make proteins, but also CRISPR RNA to function. And this RNA contains each piece of this barcode that's already in the CRISPR region. And after the expression, it forms a complex with the Cas gene, effector gene, and can guide the complex for target cleavage or immunity stage. And this is how game is over for phage. So basically, it's the basic schema how most of the CRISPR complexes works. I say most because there are some others related to transposition and so on. There's a huge variety of CRISPR involved right now. But basic schema is nucleic acid destruction. And so it's the mechanism how phage is eliminated. Why at all it is important? Actually, in reality, phage infection is quite a big problem. For a factory, for example, like cheese factory, where you have bacteria that perform fermentation, and they can get attacked with phages. And if you have, for example, cheese that needs 24 months, so if you get phages at that stage, you will lose quite a lot of money during the production process. And in history, actually, CRISPR immunity was first discovered by scientists who work in food factory, in milk factory, daily products. And after that, CRISPR was discovered. It became important for pharmacy, for genetic engineering, for organisms such as chimpanzees, for example, or human, as you know already. For Drosophila, for tomatoes, for Arabidopsis, for zebrafish, for many, many others. And actually, even in 2013, which is quite a while ago, Forbes, which is famous international economic magazine, called Cas9 protein. This protein could change biotech forever. And already we have Nobel Prize and actually huge attention towards Cas9 protein. And Cas9 is actually painted as a blob, as I say. And it's actually funny because Cas9 is painted like a blob, which recognize short sequence called PAM. PAM sequence is protospacer adjacent motif. It is short sequence that has to be close to the Cas9 target. It just can vary, can be GG, can be other nucleotides. And then these two crosses represents cleavage sites of Cas9. The RNA, it's the guide RNA that came from CRISPR region. But actually, we know that Cas9 and all other proteins are not like blobs. So I'll tell you more about Cas9. And at first, I will show some nice examples of genome editing. For me, one of the nicest examples is change of the color pattern of butterfly wings. And deletions of melanin pigmentation gene changed A-spot color pattern. I-spot is the spot on the wing. And this is called I-spot. And after deletions of one transcription factor, they removed this I spot fault. And this is loss of function. And then the other gene, called distal less, gave more spots on butterfly wings. So we got two genes, and one of the genes gives less eye spot pattern, and the other gene gives more eye spots on the butterfly wing. So it's quite a nice example showing that Cas9 can perform either this or this genome engineering task. But I have to tell more about Cas9. As I mentioned before, Cas9 came out of daily products industry, and how overall Cas9 was noticed. Scientists working with yogurt noticed that bacteria who contain extra spacers, extra DNA sequences in CRISPR region who came from different pages. For example, this or this page, if a bacteria contain extra barcode from this page, it is less sensitive to that page, comparing to bacteria who does not contain the spacer, which is here. And the other bacteria containing spacer are less sensitive to the page, so means they are more resistant to the page. And this is quite all for science paper 2007. And this was the first paper where exactly Cas9 and CRISPR activity was demonstrated in vivo. And they noticed the phenomena. And would you suggest any experiments how to understand the action of the system? The question for those who are biochemists. What would you try? What would you do to understand which components are important from this system?
Maybe they could try knockout experiments.
Yes, exactly. That's what was done later. The knockout, I'll also tell you about knockout, which is here. But at first, scientists tried to take plasmid, which contains spacer, and also the whole CRISPR region, and to see if the plasmid that contains spacer gets the same transformation efficiency as the plasmid without spacer. And they noticed that if plasmid contains target, it gets much less transformation efficiency comparing to the plasmid without target, meaning that CRISPR in unknown mechanism targets its spacer. So means that CRISPR region works as some guide. And then they tried knockouts, as somebody suggested. And knockouts turned out that the only protein which is important for interference is Cas9, because Cas9 knockout completely removed the plasmid, the ability to restrict plasmids. And in this way, the scientists showed that Cas9 is a single protein that is important to guide CRISPR region and target the plasmid. So it's many details here. So maybe I can provide slides and you can look at it after the talk, because it's all published data that I present. And then after this experiment, Cas9 is the only protein which is important for interference. So take Cas9, take CRISPR region, and try some in vivo experiments. But it doesn't work that easy. And scientists struggled for quite, not many, but struggled. And it turned out that CRISPR action needs extra piece of RNA, which is called trace RNA. And it was coded here, upstream of Cas9. And in the first experiment, actually scientists got lucky, because they took this little piece of nucleic acids here. They didn't take Cas9 right from the start, but took a little bit of genomic DNA. And therefore the system worked, even though they didn't know, but they didn't know that trace RNA is present. And the trace RNA appeared from sequencing experiments from Dr. Emmanuel Charpentier group. And this piece of RNA is important for maturation of CRISPR RNA. And Cas9 actually needs a short piece of guide to fully function. If you get long pre-CRISPR RNA, it's not functional, and it needs to mature. This trace RNA is an important factor to get single piece of guide into Cas9 protein. So therefore, actually this is the main paper by this lady got Nobel. And this result was that important. And after the trace RNA paper appeared, my colleagues from Lithuania tried experiments with Cas9, and they mixed the trace RNA, CRISPR RNA, and Cas9 protein with a target sequence that contained PAM and double-stranded DNA, and showed that the guide cleaves both strands with staggered ends. And this was not only proven by gels, but also with sequencing experiments on plasmids. And actually, it was unfortunate that this paper got retracted, and then another paper got published, which came from Jennifer Doudna. But actually, both papers are showing very similar results, as you can see here in this figure. Because here also double-stranded DNA target, the guide, and the cleavage product of the Cas9 protein, which is actually not that clear that it's that dense. But from sequencing data, it's more clear. And maybe a lot of other experiments with supercoiled plasmids and with guide RNA and more experimental data got that paper that further. And also California. And what else was done? Cas9 has two active sites, RUFC domain and HNH domain. And the scientists mutated these domains, and it turned out that if you mutate either which domain, you can have a nickase. You can have a protein that nicks a single strand of DNA. So that's the second paper, which got Nobel Prize, which majorly got Nobel Prize. And also what else was shown in that paper? As I mentioned before, the trace RNA and CRISPR-RNA are both important RNA molecules for Cas9. So in this paper, they constructed a chimeric RNA, which found guide RNA and tracer RNA together. And they showed that there is a combination of chimeric RNA that can also perform cleavage with Cas9. And now this chimeric RNA is called single guide or guide RNA. It is known as guide RNA simply by people who just use CRISPR. So we come back to the genome engineering slide. And actually, we come back to that slide because after the discovery of Cas9, it has been used in many, many different areas. And the patent for and so on was the main thing who pushed the field forward. And after that, a variety of CRISPR-Cas systems has been discovered. And Cas9 protein is actually maybe the most known protein. But there are many, many other examples of CRISPR-Cas systems that work in different mechanisms, in different targets, and that can be used in different applications. And I will shortly cover this for you, just that there is a type 1. Type 1 forms multi-protein complexes. I will show you later. And there's type 3 that targets both single-stranded RNA and single-stranded DNA. And there is type 5, which targets single-stranded and double-stranded DNA, and type 6. So Cas9 is here, as you see, is the only type out of 6 types. So there are many, many others. So I myself work on class 1, on CRISPR complexes. And these complexes form multi-subunit complexes with CRISPR-RNA guide. And they form R loops on DNA. And then there is additional nuclease helicase that can target and destroy nucleic acid. This is for type 1. Type 3 is very, very complicated. And type 3 targets both RNA, which is here RNA transcript, and this RNA transcript is targeted by complex and cleaved into pieces, which is labeled blue. And then after the transcription occurs, it also can target single-stranded DNA, maybe in the RNA polymerase complex. So this is very complicated complex, and it has also an additional activity. It cleaves, as I said before, it cleaves the transcript, it cleaves single-stranded DNA, and then it synthesizes a very unique signaling molecule called cyclic adenylate. And the complex, after it binds the target, it synthesizes cyclic adenylate signal. And this signal is not useless. It's used for activation of non-specific ribonuclease, CSM6. And this non-specific ribonuclease can target, for example, phage RNA. So this CSM6 is called auxiliary CRISPR protein. It's not necessary for CRISPR activity, but it helps to fully perform the activity. So this is the main field of my interest, auxiliary CRISPR proteins. And I will just cover a bit my latest paper, where we discovered toxin-antitoxin system in the same operon as Cas genes. And this toxin-antitoxin genes maybe work to cooperate with CRISPR, as we suspect. There are two genes, which is antitoxin and toxin. And if there is enough of ATP in the cells, the antitoxin is switched off by novel covalent modification, deadenylation or deampylation. And if there is not enough ATP in the cells, ribonuclease is active and can cleave tRNA. And it removes tRNA 3'N, which is important for amino acylation. And what we showed in the paper, that this modification interrupts between two helixes and change the active site loop, change the conformation of the active site loop, and this loop gets turned off, gets in inactive conformation. And this is our crystal structure, and also this is very complicated slide. The main message of this slide is that it cleaves tRNA. And actually we needed quite a lot of experiments to prove that, because it was different from the results of other groups. And therefore we had to perform many experiments to prove that really tRNA is the target. So this is also an extra mechanism how CRISPR can cooperate with toxin antitoxin, most probably. But we just know that this cycle works, but what else, we don't know. And also other molecular tools. Cas9 lives with blunt ends. There is also Cas12, which leaves staggered ends on DNA, and is also used for genome editing. And also Cas13, which is used for RNA cleavage. It uses target RNA to activate non-specific nuclease. And this method has been used for nucleic acid detection method called Sherlock, which has been employed even for SARS detection. So the method works in the way that it grasps RNA out of the RNA mixture. If the virus RNA is present, then the Cas13 is activated, and it non-specifically cleaves thread probe, and the signal is released. So if different Cas13 proteins are applied for this method, mixture of different viruses with different Cas proteins can be detected. Like Zika, dengue viruses can be detected by this method. So that's kind of it I wanted to show you. And I want to thank my supervisor and my university for financial support. And thank you for your attention of this talk. And if you have any questions, I would like to hear them.
Okay, thank you. In the chat.
Just a little louder.
Did I interrupt anyone saying something?
No. Ask.
I was just going to say that there is a question from Vidmanta in the chat, in the group chat.
In the chat, okay. What CRISPR systems are going to be applied to humans most likely? I suspect that Cas9, because Cas9 is already a mostly studied protein. And after that, I think Cas12, because Cas12 is similar and it leaves staggered ends. And it's more, it's better for homologous center combination. What else for RNA editing Cas13? Because RNA editing is also emerging field. And also type 1 systems are already applied for human as well, for human cells. They just are fused to nuclease. For example, FOC nuclease can be fused to cascade complex and then targeted to the genome. There are many different ways you can play. What problem could arise when applying this CRISPR system to humans? Of course, the main issues are ethical issues, and there are many discussions regarding whether it's allowed, it should be allowed to apply, or in which case it should be allowed, in which not, and so on. But I think this is the main problem, that we don't have any law that already defines how to use genetic engineering. And it's already emerging, but the scientists working with it get somehow defined, or how to say, we should try it in order we want to know how it works. But how to try it? We need to have restrictions, we need to have law to define it. So it's kind of, is it safe technology for gene therapies? There is no technology yet which is fully safe. And I wouldn't say that it's fully safe, because of targeting effects, they are possible. If you target your target, there is even small probability that you will get off-target effect. So I think there is no technology yet which is fully safe.
I was also wondering, you mentioned about CRISPR RNA maturing, and I don't quite remember when that RNA is said to be mature.
It is mature after it hybridized with trace RNA, and after it is cleaved with RNase 3, which is, I didn't mention this RNase, but you need the host RNase to cleave this and then if you have a single piece of spacer and trace RNA, it's already mature. Is it fine now?
Yeah, it's better when I remember. So thank you. And also, I wanted to ask actually about not about scientific point, but about your group. Like, as far as I remember, a few years ago when I did an internship in Life Sciences Center, I heard many people expecting Professor Shikshnys to get Nobel Prize for investigating that CRISPR-Cas system. So why do you think, Nobel Prize can be shared by free people? So why do you think that your supervisor or your professor was not included together with the two women who won the Nobel Prize?
It's actually, I don't know how to answer. Maybe it depends from publishing raw as well. Then the first paper which is published was not from professor, and maybe also I would say that Jennifer Doudna, she is famous for many other things like ribozymes, or she's very well-known scientist, and maybe that's why also she draw more attention. And actually I wouldn't say that we are definitely worth to get that Nobel Prize and we didn't get it. But overall, the paper, which is showing the same results, it's a proof. But I would say that my colleagues were disappointed that the day the Nobel Prize were announced, and we didn't talk about that day even with them.
So are you saying that the Nobel Prize that was won for that was won due to relevant papers on CRISPR-Cas system actually is not just for that, but also for that scientists having a relevant addition to other topics in science too?
I respect both of them, actually. I respect both Jennifer and Emmanuel. They are both great scientists. And they contributed much to the field, as well as others who didn't get the prize. The prize can be only 3 people, but there are, if you include, for example, Feng Zhang, which developed many essays. For example, Feng Zhang developed eukaryote, the first eukaryotic use of Cas9. So why we don't include that person, and so on, so on. We can count many scientists who developed that technology, but I think it's also how much known you are in the field. How are you respectful in the field? It's many, and it depends also from the country, from the politics, from the attention to scientists, from the public well-known names. But I don't say that it's sad that we didn't get Nobel Prize, because at least we have attention anyway to the field. I mean,
see.
I look at it as a positive sign that the field got the Nobel Prize, not a sign. Even though I respect both of them, they are both great scientists.
I see. Thank you.
Vidmanta doesn't look happy.
No, I'm happy. I just thought before that Nobel Prize is given for one specific discovery. So I think that, well, it's not an argument that that one woman won Nobel Prize because she had contributions on, you said, ribozymes, or I don't know, another thing.
Yeah, ribozymes. She has a
Yeah.
lot of contributions.
Oh, so it relates to CRISPR. Oh, so it's fine then. Yeah. So I think
It's
just
not
her
very related to CRISPR.
No?
No. It just she's well known in RNA world, in ribozymes. She's very well known as a crystallographer.
Yeah,
She
so when I
knows
just
how to crystallize, for example, ribozymes. Book chapters at least.
Okay. That's interesting. When I got to know that Nobel Prize is not given just for discovering one specific thing, but it's like for having just contributions to many fields, not just to one field.
It was not mentioned that it's for other contributions. It was mentioned that it's for Cas9, but I think it depends if you are a well-known scientist for other things.
Okay, thank you very much. So do we have any other questions? If not, I think that was a very well-prepared presentation with many data. And if somebody will want to look a bit in more depth in this CRISPR-Cas system, we have all relevant papers, and it's much easier now to get deeper by knowing those relevant scientists and just to get better
Yeah, we have a special
understand
course.
CRISPR-Cas system.
We have a special course at university called CRISPR-Cas technology for master students. Yeah, and we engineer bacteria. We improve dairy products to get more resistant Streptococcus thermophilus. And that's kind of it. But it's enough for half a year.
I also wanted to ask your opinion about the experiment that was conducted on twins by a Chinese scientist. Like, what did you think about that? I know it's kind of personal. I might turn off the recording if it's too controversial, but if
it's actually quite strange that they chose those two genes, those receptor genes,
yeah,
for HIV. I don't know why, but yeah, it's controversial experiment, and I don't know. Right now, it maybe those twins have some advantage over others because they cannot get HIV, but
from what
I don't know how much is that of advantage.
of the outcome of the experiment, but I'm not sure. Maybe
But right now, if we want to check, we have to infect them actually, which is not also
yeah.
very good. It's like experimental animal, experimental human.
What did you think of the actual sentence that's been given to the doctor, to the scientist? Because I found it really controversial in a sense that, the study was performed and approved. So it wasn't like specifically the fault of the scientist who conducted this, rather than like it should be kind of spread between all of the people who were involved in this. And it was really interesting to see how to observe how the whole situation kind of unwrapped.
Yeah, for me too. It was kind of weird, but I don't know actually what's the situation right now.
I think
Do anyone know what's the situation right now with the scientist who conducted genome engineering on twins?
I think he was put in prison, but I'm
Yeah, and I remember he was put in prison, but after that he has to be somehow. I don't remember actually what's after.
it would be interesting whether he would be allowed.
Didn't we have to pay? I think we had to pay some money to the family of those twins, if I remember it correctly.
I think yes. I'm reading the article about it now. Yeah, they had to pay compensation.
But actually the conference on which he presented was also, he presented it and all of the community like got crazy.
Yeah. It's unlikely that he will be able to work again as a researcher, like in any kind of institution or university.
Yeah, it said so. But is he still in prison? I don't know.
I'm also not sure. I think he's. I don't remember the length of the sentence.
Two collaborators were fined. Yeah, it can be.
Oh, okay. For 3 years.
And fined.
I just wonder whether there was some consequences for the institution, like for the university he was working in, or did they just?
Yeah, it should be because at least it's not a scientist
Yeah.
actually who is conducting the experiment. It's actually funding agencies and so on. Many people behind should be there, which is not mentioned here.
And in terms of your work, you mentioned that you apply CRISPR technology to dairy products, or do they?
Not exactly. We apply as we do our lab work in university.
Mhm.
But overall, I work on fundamental side. I try to find another molecular tools,
Mhm.
basically multi-protein complexes that can also cleave DNA and can be used also similar as Cas9.
Mhm.
But so far, no luck.
And is there any chance that we will ever get to cleaving and editing not just like one or two nucleotides, but multiple sites at the same time? Because I've read that off-target effects for multiple nucleotides are really high, and that makes it not feasible for gene therapy in that instance.
You mean multiple nucleotides like pieces of DNA?
So
Or
when I was like 2 years ago, I was doing work on comparing the genetic tools to target genetic disorders, like why gene therapy, and like it was kind of a low level because it was still not even university level coursework. But I remember reading that it's only feasible to do this for genetic disorders which only have mutation at like one or two sites maximum. But for all of the other genetic disorders, the off-target effects of current CRISPR tools are too high.
yeah, it's kind of true, but maybe in parallel that can be applied. I mean, in rounds,
Oh, okay.
these tools can be applied. And also these tools can be applied for detection of mutations. If you knock out both sides in Cas9 and you get a fluorescent label on it, you get a detection tool.
Yeah.
And then you may detect some point mutations in some regions that can be important for diseases. Also, of course, sequencing can also be used for that, but these tools are easier if they are developed. If they are well developed, they are easier to use.
Thank
So
you.
far, I work on
There's also
cyanobacteria
a question on the chat.
on the chat. Okay. Relating to, I was wondering if the magnetic field superpole assay was developed especially for this study or whether it was other applications. It seemed like a pretty smart solution to understand the loop formation, but did it originally have different use? Twitter assays originally were mostly developed to understand how helicases work. Helicases are the proteins that unwind DNA. And also, this is a good way to understand the loop formation. And also, the Tweezer assay can be used to monitor nicking. If you get a super coiled plasmid and you get a magnetic bead on it, and it gets nicked, you will observe that it got nicked. The bead will get up. It's very specific, actually, question. But no, sorry. Why you say sorry? yeah, it's a single molecule assay. It's not me who developed this assay, but I applied it for CRISPR. Wasn't sure it was a bit of stretch from the original material. Yeah, it was a bit. Yeah, I was more talking about my latest paper from toxin. From toxin side.
Any more questions? Okay, so thank you very much, Ben, for a wonderful presentation. And
yeah, I can share slides with Vedvante and then you'll share with the other group.
Yeah, would be great.
Yeah, thank you for listening and thank you for discussions. And thank you for organizing the meeting.
Thank you so much for coming and for giving such a wonderful talk.
Yeah.